All this is Dr. Mobeen Sayed. Welcome to a cafe chat. And um, there had been some protest last time that I was just teaching and not chatting. So today we'll chat. So let's start. How is everyone? Jean, welcome. So I do want to quickly run by you a study that I read a few days ago as well. A KoolBean Bill also sent that over. So just a very quick review of that study. I think you'll like it. So this is drbean.com. There are many more lectures here for medical education. And this is the study. Adipose tissue retains an epigenetic memory of obesity after weight loss. So I'll just go over some parts of the abstract and nothing more. We'll discuss that in detail later on. But generally, what they're saying here is that if somebody has gained weight, the adipocytes, the fat cells, they develop a genetic change in them, epigenetic change, meaning the changes that occur in DNA because of environmental factors. So here the environmental factor being fats or fat collection in the fat cell. So the fat cell then remembers those changes. And even when the person loses weight, these fat cells still continue to misbehave. They still stay insulin resistant. They still are metabolically deranged. They still would tend to quickly collect fat. And that is why the rebound from losing weight to gaining weight again becomes easier because the fat cells are already primed and they stay primed. They forget that the person has become lean. So they bounce it back. So here very quickly, reducing body weight to improve metabolic health and related comorbidities is a primary goal in treating obesity. However, maintaining weight loss is a considerable challenge, especially as the body seems to retain an obesogenic memory. The body seems to retain an obesogenic memory that defends against weight changes. So the cells actually learn to stay obese or to stay fat filled and to behave in a way that they fill with the fat, for example, being insulin resistant. And so they just keep remembering that. Overcoming this barrier for long-term treatment success is difficult because the molecular mechanisms underpinning this phenomena remain largely unknown. Here, by using single nucleus RNA sequencing, so they take a single cell's nucleus and see what kind of RNA is coming out of it. We show that both human and mouse adipose tissues retain cellular transcriptional changes after appreciable weight loss. Furthermore, we find persistent obesity-induced alterations in the epigenome of mouse adipocytes that negatively affect their function and response to metabolic stimuli. Mice carrying these obesogenic memory show accelerated rebound weight gain, and the epigenetic memory can explain further transcriptional deregulation in adipocyte in response to further high-fat diet feeding. In summary, our findings indicate the existence of an obesogenic memory largely on the base of stable epigenetic changes, so the changes in the DNA due to environment, which stay there even when the person loses weight, in mouse adipocytes and probably other cell types. These changes seem to prime cells for pathological responses in an obesogenic environment, contributing to the problematic yo-yo effect often seen with dieting. Targeting these changes in the future could improve long-term weight management and health outcomes. So very interesting, fascinating study. So I'll go over this at some other time, but here we are. Marcy says, missed your awesome sense of humor from a while back. Thank you very much and welcome. Slam RN says, can anyone see me? We can all, I think, see you. Now, anybody who will watch the video can see you too. Casey says, good night. Good night, Casey. Take rest. Okay, so what else is happening? Sorry to Alquin for your cat and for your health. Luffy had been sick as well for some days. He went out at night, got bit by another cat, developed an abscess. We got the abscess drained. Then we kept cleaning and giving him antibiotics. It's very difficult to feed him medicines. My net, it is like a clockwork. Every 6 or 5, 10, my net has to go down once. So anyways, so then he developed the abscess again. We went out again. We got it drained again. Then he stayed on the antibiotics for 2 weeks. So now it is 10 days after or more, and he is okay. So I'm hoping it is behind us. But he is grounded for for going out at night. BR says, glad you're doing these chit chats. Thank you very much. Glad you are here. Joanne says, poor kitty, you need the bullseye pill gun for Luffy. Okay, so I'll have to see what that is. They gave us, we told them he doesn't take pills at all. Even if you, mix them with treats, he will detect the pill and throw it out. So they gave us liquid forms, and we had to, fill those syringes and then open his mouth and and feed him through that. And he didn't like it. He would injure me, my wife, with his, claws. But I'm happy it is behind us. Slam says, cats are so smart. Correct. So what are we talking about today? Today, it's your turn. I will not go into the details of teaching. So you tell me, what do we talk? Or do we stay quiet? If so, Alquin says, GLP-1, how is it so miraculous? It's a very good question. So let me actually open up something that we can look at. Okay, so I'm going to go into some discussion of GLP-1. What happens is that so the first thing that we have to understand is that there is a thing called intestinal break. What that break is, that when we are eating food, of course, the food will go through our mouth to esophagus, then to stomach, then duodenum, jejunum, small intestine, ileum. Then it will start filling the colon, and finally, some of that food will become fecal matter and be removed. So what happens is that as we are filling, we are eating, there is some previous food that, let's say, we had it at breakfast or we had it at lunch, and now we are eating dinner. The previously traveling food is already now filling the colon area. The newer, relatively newer food, but again, a couple of hours earlier, that is filling the small intestine. So what these intestine do is that they send a message to the upper areas, including stomach, and to the nervous system, and they tell them to give it a break. The lower intestine tells the upper intestine and the brain to say, can you stop eating? Can you stop this human from eating? because we are already full. So this is called intestinal break. The most important effect of that is on the stomach. Intestinal break, or the GLP-1, is released from the intestine, and then so it is the hormone released from the lower intestine, which will work on the upper intestine, especially the stomach, and reduce the acid secretion in the stomach. Why? Because when the stomach is mixing acids with the food will break down, quickly move forward, and start getting absorbed. So when you reduce the acid secretion, the food will not digest that fast, and it would stay in the stomach for longer. The second thing is that the stomach's movement becomes reduced as well. So when the stomach is not moving as fast, and when it is not emptying as fast, then the higher centers, the nervous system, which is connected to the whole GIT, that nervous system will get a message that it seems like I am full. The stomach is full, so the human is full. And the nervous system would create satiety, and it would satiate us. It would take our hunger away, the feeling of hunger, and we would eat less. That is a basic mechanism. So what happens is folks who are on GLP-1, they would actually see the same food that they have been eating before, and their stomachs are not moving, the stomachs are not emptying fast enough, and they would look at the food and they will not want to eat it. So the hunger reduces. This is one effect. Secondly, even when they eat, the stomach doesn't digest very well. The stomach does not move the food very well. So they start developing those burps, and they start developing that feeling of fullness, which when the food becomes a little stale, it becomes a problem with the feeling. And so the person just becomes averse to eating. That reduced hunger then causes the weight loss. And they did a study on women. I think these were a specific category of women. They looked at their stomach emptying. They compared them to control group who were not on GLP-1s, and these women were on GLP-1, and they saw that the women who were on GLP-1, even 4 hours after eating, their stomach still contained food, compared to the control who were not on GLP-1, who had their stomachs emptied. So imagine food sitting in the stomach for 4 hours. There is a feeling of fullness, but on top of that, the food is becoming a little bit stale as well, and there are burps, and there is a discomfort, and that makes the user a little averse to it. Of course, what happens is that when GLP-1s are given, they are given in a stepped dose. They're slowly increased, so body keeps adapting as well. The other interesting thing about GLP-1s is that they have a direct effect on the medulla. So brainstem, so the spinal cord goes up to the skull level. Then from there, the brainstem continues, and then the brain. Brainstem has an area which is called chemo trigger zone. So chemotactic trigger zone, I believe, or CTZ. CTZ, when that area is stimulated, it produces a feeling of nausea and vomiting. Interestingly, GLP-1 have a direct effect on that area, on CTZ of medulla, which is part of the brainstem, to produce the feeling of nausea and vomiting. And that is a direct effect. There were many theories before where people thought, or the researchers thought, or the doctors thought, that as the food is full in the stomach and the stomach is not moving well, that is what causes the feeling and the perception of nausea. But they found out that the GLP-1 can actually directly act on the medulla to produce nausea. So nausea is a direct effect. In addition to that, GLP-1 can work on the vagus nerve. So the vagus nerve is the main nerve, the parasympathetic nerve that is innervating many visceras, including GIT. GLP-1 can actually trigger vagus nerve as well to affect the brain for not eating more and for this perception of fullness and nausea. So this is a very interesting drug. When given in a stepped dose, it can actually be very useful. And as it has been, you have seen many Hollywood actors and personalities becoming slim very quickly. However, nausea, vomiting is one side effect, which is actually a direct effect, not a side effect. For example, stomach not moving. Stomach fullness is another side effect, and the acidity, lack of acidity causing food to be stale. Third is the pancreatitis. In some patients, it causes severe pancreatitis, and that can become a problem. And I've seen or heard from patients that even when the dose is reduced, they still develop pancreatitis. Normally, they say that if the dose is reduced, then reduce the when the pancreatitis or the pain appears, reduce the dose, try again. It also has an effect on the thyroid to cause cancer, or it is associated with thyroid cancers as well. And something that is very interesting for me is that it can cause constipation and paradoxical diarrhea, or diarrhea that is called overflow diarrhea. So that is a very interesting mechanism. So let me explain how that mechanism works. Let me just very quickly okay, so there is some other discussion going on as well in there. But let me explain how the overflow diarrhea occurs. So imagine that, let me see if I can make it a tiny bit bigger. Somehow let me actually close this, start this program once more. Do you know that this drawing board, I wrote it by myself in C#. So it is my own engineering code. And if you see why I did that was there are no interfaces. There is nothing here. And I just wanted to have just a whiteboard. So everything else is with the keyboard. For example, I can change the color with the keyboard. I can change the size with the keyboard. And so I wanted everything to be with the keyboard. Anyway, so here is what happens. Because the GLP-1 reduce the mobility of the intestine as well, what happens is that the patient starts developing diarrhea. So let's say this is colon, although colon is not like this. Let's make it a little more accurate. So let's say this is colon and this is rectum and then anus. Let's say this is transverse colon. This looks like a megacolon, so don't mind. So as the mobility reduces, that allows more time for the developing fecal matter to stay in the intestine, in the colon, and more time for the water to be absorbed from it. The longer the fecal matter or the stools stay within our GIT, the more the water is absorbed. So when the Alcuin says megacolon what? So megacolon is a pathological condition in which there are some children who are born with lesser neurological innervation in these areas of colon, rectum. And what happens is because they do not get the sense of their filling, because the nervous system isn't working there, they would just keep having fecal matter collect there and not be ejected. And so their colon would just keep expanding. And so that is called megacolon. It is a very painful and bad situation for the child. So back here, as the food stays here, food or the fecal matter, developing fecal matter, that stays here for longer, the water is absorbed from it quickly, and this becomes hardened stool. So the hardened stool would cause constipation. Now, think about it. This is a very interesting thing. Constipation is now the problem. However, the patient is still eating and more food is still coming in. And because the intestinal system isn't absorbing very well, because the digestive enzymes are not working very well, a lot of more fluidy, liquidy food reaches here. And that food, let me make it in blue color. This food that is reaching here is more liquidy. So what it will do is it would find its way around the fecal matter. And from there, this liquid would go and come out of the anus. So now the patient is constipated, but also has diarrhea. This is called overflow diarrhea. And GLP-1s can cause a side effect of overflow diarrhea as well. So of course, their first side effect is going to be constipation, and then overflow diarrhea can occur. And then there are a bunch of other side effects too. But this is how the GLP-1s work. They work by intestinal braking system. They work by causing medulla to produce a feeling of nausea. They work by reducing hunger. And that is how they help with the weight reduction. Yeah, sigmoid colon, correct. So we have the descending colon, sigmoid colon, rectum, and then anus. Who said that? Jim said, don't forget the sigmoid colon. Absolutely. I drew it, I didn't say it. So So Nipa says that ferroptosis and C15. So Nipa, I'll talk more about it, but just generally, ferroptosis is, look, these cells, when they die, so let me even, before we say cells die, so let's say this is a happy little cell, right? Just like Bob Ross's happy little trees. This is a happy little cell. And it is doing its function, whatever its function is. It's a liver cell or a pancreas cell or muscle cell or something. It is doing its function, it is happy, and then some stress appears. That stress can be from within. For example, as I've been discussing, that the cells that are getting a lot of reactive oxygen species, or the cells that are receiving a lot of glucose and they are making a lot of misfolded proteins as a result of reactive oxygen species production. We have done this discussion before. So internal stress or external stress. That stress is managed in multiple ways. The first thing that the cell would do is would try to trigger autophagy. It would try to clean up itself. Plus, whenever a cell is under stress, the cell thinks that I'm soon going to be deprived of nutrition because I have this stress on me. And if there is this stress, then it is possible that soon after there will be some sort of nutritional problem as well. So the cell triggers autophagy to eat away any garbage that is present in the cell, or in the medical terms, misfolded protein aggregates. So that is the best outcome. If that works, the cell will actually feel healthier, better, and it will be able to cope with the stress. In addition to that, so let's call it 1A, the cell also activates heat shock proteins. Now, the name is heat shock proteins, but really we should call them stress proteins. Whenever a cell is under stress, it produces these proteins, which will then help clean up the cell by even triggering the autophagy. Plus, the heat shock proteins do a bunch of other things. For example, they provide an incubation space. They act like little incubators where damaged proteins can get in there and be protected from the hostile environment outside, and they're given a chance to become corrected again. So the HSP provide that facility and try to heal the proteins that are disabled or that have been damaged. Even a cell performs better than us, human community, that if we have folks who are not doing well, especially, for example, in vaccines. So the cell actually activates these heat shock proteins, which would then provide a safer environment and protect these proteins from further damage and allow them to heal. Heat shock proteins also pick up the damaged proteins and bring them to endosomes, which are recycling systems of the cell. And they would pick up these proteins and kind of recycle them. And so these are like wood chipper, that on one end you insert the proteins, and from the other end the broken pieces or the amino acids would come out, which will then be recycled to make other enzymes, etc. So this is the cell's response to stress. And there are many other stress responses. I'm simplifying it to reach this point that if autophagy and heat shock protein system is not able to remove the stress or alleviate the stress, then the second process that starts is autophagy. Sorry, this autophagy, that the second one is going to be the one that is more, it would start even eating away at the cell's own organelle, for example, Golgi apparatus. So autophagy is of two types. One autophagy is that would heal the cell by removing the misfolded protein aggregates, and the other autophagy is damaging or apoptotic autophagy that would kill the cell because it would start eating away at the normal organelle of the cell that are doing various functions. For example, Golgi apparatus, endoplasmic reticulum, peroxisomes, mitochondria, etc. So this autophagy would be started as well. Now, in this meantime, cell, if the stress is still present, then the cell would say, you know what, I have done everything that I could, and it is time for me to die. And that is called apoptosis. Now, some cells in this state will decide that I'm not going to die, and instead I will become an exhausted cell. For example, immune cells do this very often. Here, they're not dying, but they said, I am just going to sit here. I'm going to do nothing. I'm going to be all grumpy, and I would continue to release inflammatory molecules, which will be my signal that I'm unhappy, I'm unwell, I'm not feeling good. When the cells start apoptosis, which is a healthy way for a cell to die. Why is it a healthy way? Cell is not able to handle the stress. Cell knows that I might get damaged very soon. And if I do get damaged, then the damaged proteins and RNA and DNA, my guts will spill out, and the immune system will become all upset and inflammation would start. And the cell says, you know what? I do not want inflammation to happen. I do not want other brethren and sisterin cells to die. So why not I just silently die away without causing inflammation? So that is apoptosis. If the cell is not able to conduct apoptosis successfully, it will also become exhausted. An exhausted cell is also the one that was not able to kill itself successfully because the killing machinery did not work good as well. Whatever damage was going on within the cell, Even the autophagic system and apoptotic system were not working correctly. This cell is going to become exhausted as well. It would just start releasing various kind of inflammatory molecules. The good news is that we can actually restore these cells, exhausted cells, back to normal, and the cells that are trying to do apoptosis back to normal if we reduce the stress in the cells. But we are going to something else. In some cases, the cell does not take the pathway of apoptosis or silently dying or physiologically dying. Instead, the cell takes a route of pyroptosis. Pyroptosis is where cell knows I'm going to die, but before dying, what it does is it produces a bunch of inflammatory molecules. It manufactures them full speed, produces them big packets. You can think of those as big bombs. It manufactures them, fills itself with those inflammatory molecules, and then activates a system that ruptures the cell itself. So then it ruptures itself. For example, there are proteins called gasdermins. They would open big holes in the cell. And from those holes, these inflammatory molecules would come out and cause havoc in the areas around. This is what is called pyroptosis, or the death by fire, or fiery death. What are those pyrotechniques for the fireworks? Jim says, so heat shock can fight cancer. They do fight cancer. Heat shock proteins are very important for fighting cancer. The problem is that the cancer cells would try to modulate the heat shock or the cell's defense mechanism. Cancer cells turn off the autophagy systems. The cancer cell turn off the apoptotic systems. Cancer cell turn off these defense mechanism where a cell would prefer to just die away. And they would say, no, you're not going to die. We are a cancer cell. We are not going to die. We are going to turn off all the defensive mechanisms and live on. And that is how they try to come out of it. Alcuin says pyrotechniques, correct. So pyroptosis, Eugene says, not for the first time. I appreciate that they made me learn ancient Greek in high school. I wish I learned that too, and I'll understand these terms better. So pyroptosis is the fiery death. Fiery death, not only the cell itself is dying, but it would cause so much inflammation around this that the tissue is just going to be under lots of stress because of all of these inflammatory molecules that are now released. These molecules would cause the nearby cells to become activated and start having the apoptotic ideas. And then these inflammatory molecules would activate the immune system, and immune system would just go berserk and start killing the cells around. And so the tissue damage would start occurring. This is the cell's revenge, that you put stress on me and you did not let me heal. Now I am so angry that I'm going to now kill a bunch of other cells while I'm dying. Then there is another cell death that is called necrosis. In the necrosis, this is more of an accidental cell death, not much planned. In the necrosis, what happens is that the cell all of a sudden does not get the nutrition that it needs and the oxygen that it needs to function correctly. So it was a healthy, normal cell. And all of a sudden you cut the nutrition, no nutrition. For example, when a coronary artery is blocked, all of a sudden blood supply to the cells downstream is gone. The cell doesn't have nutrition anymore. The cell doesn't have oxygen anymore. The cell cannot wash its trash that it is producing, the lactic acids and the creatinine and other damaging molecules that do not go away. And this cell just dies because of that accident that happens to it. So this is necrosis. Then there is this ferroptosis. Ferroptosis is a more recently, by recently, I think 2006 or so, is when this mechanism was found. In this mechanism, the cell that is planning to die would allow a lot of iron to come in and accumulate in the cell. Luffy is, so it is raining outside. I have opened the door for Luffy so he can go out, but he won't go out because it is raining. And he is just now upset that why can't we stop the rain and he can go out. So this is Luffy is the brat. So this is ferroptosis. A lot of iron is brought in, then that iron starts interacting with the cellular machinery and starts disrupting their function. And now the cell is kind of in a simple terms filled with iron and it dies. This is called ferroptosis. So there are various kinds of methods for a cell to die. The best outcome for a stressed out cell is autophagy and healing back to normal. But continued stress will make the cell not be able to withstand and try to do uh, apoptosis. First, it would do autophagy, it would activate heat shock system, then it would do apoptosis towards healing. If it cannot do, autophagy towards healing, then it would do autophagy towards even trimming its own organelle. Then it would try to die by apoptosis. If it cannot die, it can actually do pyroptosis, or it can become exhausted cell. And necrosis is an accidental cell death. Cell got crushed, for example. And ferroptosis is also a pathway. Cynthia says Luffy wants Dr. Bean to go in the rain. So I have been opening front door and back door for him, and he would go out and get wet and come back and then run out of the other door. So now my wife is not home, and so nobody's opening the front door for him, and he is just upset. So this is the, in a nutshell, I didn't describe the ferroptosis to the length, but I wanted to create the big picture for how a cell behaves. This is why, if you have been listening to me for some time now, you know that I'm a big proponent of reducing inflammation, controlling inflammation. So when people say that I would muscle through the inflammation, I have a very good friend of mine who became injured, and he was not even able to pick up his legs and put them in the car. For example, once I went to have coffee with him, and he came out and he was trying to sit in the car, and he had to pull his legs up to sit in the car. And when I said that, why aren't you taking any medicines? He said, no, I would just, so he is a boxer and a, hard headed. So he said, no, I'm just going to muscle through this. And I said that this will cause damage to your tissues. You have to control the inflammation. So fortunately, I sat down with him. I explained a bunch of things. A year later, he's back to boxing. But before discussing this with me, he was in the COVID lockdown, so we were not able to meet, and he had been suffering for 2 years. And then after meeting, within a year, he's back to boxing. And now he's back to working and is fine. So continued stress isn't healthy for cell. So even when you psychologically feel that I can muscle through these things, cells would need help. And the help is reduce stress. If you can reduce stress by lifestyle changes, by psychological aspects, great. If not, do whatever to reduce inflammation. So Dwayne says, are certain cell types more prone to pyro? So maybe, but it is really the status of the cell. That is the cell given a chance to perform apoptosis? Or let me say, if a cell is given a chance to do autophagy successfully, and so you could say that, what does it mean that it cannot do autophagy successfully? So I'll give you this example. Imagine this room is a cell, and I am one of the enzymes here whose function is to speak. And for that, I need to turn on the lights and have the mic and walk through the door and come sit down at the table and do this at my chair, and then on my chair at the table and talk. Imagine that this room is so filled with misfolded proteins the aggregates, that I cannot even walk in the room correctly. So now imagine if I was an autophagy enzyme system, a worker who is going to clean up. But if I cannot even move, then I cannot clean up. This is why we should not allow our cells to reach a point where they can't even trigger their autophagy anymore. So if the autophagy fails, or if the heat shock protein systems fail, and how does the nucleus know that these are failing? Because there are sensors that are continuously sending the stress signals to the nucleus. So nucleus is now trying to open up the autophagy systems, the genes, and they're sending messages that, hey, start the autophagy, but nothing is happening. Stress signal is still coming in. Then the nucleus would say, all right, fine, I'm going to do apoptosis. I'm going to die. And if the apoptotic machinery, I mean, it is not that the nucleus thought I'll die and the cell would just die. You have to activate a number of enzymes that will have to start going and destroying things within the cell in a very methodical way that inflammation should not occur. They would flag the proteins that are being destroyed for recycling and saying this is safe. We did it intentionally. If that apoptotic system doesn't work correctly, then exhausted cell, very common in immune system cells, or pyroptotic cell, very common in the tissue cells. So I think it is not a type of tissue that would have one or the other, but the status of the tissue. So this is a very good question. Granny Page says, what, Dr. Bean, in your opinion, is the best dietary anti-inflammatory? This is a talk that I will now go on till the morning. But some ideas, just general ideas. So let me start from dietary anti-inflammatory. Omegas are the first and foremost to correct the balance of. And I have done this discussion on the other channel that it is not necessary that in every single case of inflammation, we start correcting the omega balances. Imagine somebody has a healthy body and they got infected. Now their body is going to use a lot of omega-6 to make arachidonic acids to create inflammation so that the pathogen can be attacked and destroyed. In this case, trying to interfere with that and say, you know what, I'm going to reduce omega-6 and increase omega-3, which would counter this system, that could be counterproductive. Unless that inflammation is so rampant that it is not just killing the pathogen, it is just exacerbating the tissue damage as well. So then omegas. Similarly, the best area for omegas, for example, are those who have subclinical inflammation or chronic inflammation. What is the difference between them, subclinical and chronic? Subclinical is there is continuous inflammation going on, but we are not aware of it. There is nothing sufficiently damaged yet that we have the symptoms. We have not become diabetic. We may become pre-diabetic. We haven't developed hypertension. We haven't developed renal impact. We haven't developed a lot of cognitive damage. So our body is still healthier. Our heart is healthier. Blood vessels are healthy. But the internal inflammation is just chipping away at the systems. This is called subclinical inflammation. How do we know about that? It can only be detected by CRP or interleukin 1 or other tumor necrosis factor alpha by various kind of inflammatory labs. From someone who has subclinical, imagine that they are fortunate that they got the labs and they found out they have inflammation. Or someone who has chronic inflammation. Chronic inflammation will mean inflammation that is continuous but is now causing symptoms as well. That means it is causing enough tissue damage that the results are seen, diabetes has happened, cognitive decline is occurring, GIT isn't working correctly, the inflammation in the GIT is happening, the skin isn't working correctly, the liver isn't working correctly, muscle fatigues are occurring, joint pains are occurring. Now we have a uh, knowledge of inflammation happening. In these kind of situations, first thing to correct should be omegas, because omega-6 gives rise to arachidonic acid, which is the parent of inflammatory systems. Of course, there is nuclear factor kappa B. Of course, there is TLR, MAP kinase, and NLRP3. There are JAK-STAT system, hypoxia-induced factors. There are many inflammatory pathways, hundreds of the pathways. But this arachidonic acid, you can call it the king of these pathways. And arachidonic acid comes from omega-6. So the first thing is to improve the diet, to reduce omega-6s and to increase omega-3s. If somebody is healthy, no inflammation, healthy, they can take omega-6 and omega-3 in a 4:1 ratio and they're fine. Somebody who's chronically inflamed, asking them to take a balanced omega-6 and 3 is just kind of wrong, because we are already seeing that their inflammation is more. Asking them to take more omega-6, for example, hey, for every single omega-3, you should take 4 omega-6s. No. They need a lot of omega-3 now. And that could be fish oils, that can be the other. What I've seen is that the grains and the seeds, they have omega-3 in them, but you have to eat a lot of those, almost impractically lot, to be able to have sufficient omega-3s. So fish oils, and there are some organic fish oils as well. I've done these discussions on the other channel. Even the vitamin A toxicity, that is a very common myth. I've discussed that as well, that there are probably just 4 cases of vitamin A toxicity in decades. And even those were where vitamin A was separately given, or the foods with vitamin A were given, and then blame was on the fish oil. So the first thing is to correct omegas. Second, curcumin is such an important anti-inflammatory, just like axanthocyanin is another which is very, very good. Curcumin is very good. NAC, antioxidants, vitamin C and E's combination, or NAC, or glutathione. So glutathione direct administration is very poorly absorbed. So NAC is the best way for that. So foods, EGCG, so the green tea, for example, that is a very good anti-inflammatory food. Quercetin, present in the red colored fruits and vegetables, bell peppers, red onions, other things. The berries are amazing with the antioxidants. So antioxidant, both lipid soluble antioxidant, for example, vitamin E, and water soluble antioxidant, for example, vitamin C, have to be given in combination. One cannot say that only give vitamin E or only give vitamin C and they are antioxidant. Why to give both? Well, we have lipid environment in the cell, which will not allow a water soluble molecule to enter there. Similarly, there is a watery environment within the cell, which would not allow a lipid molecule to enter there. So for antioxidation of lipid environment, lipid soluble antioxidants are needed. For example, quercetin, for example, vitamin E, for example, alpha lipoic acid. For the water soluble, vitamin C, then glutathione, and some other. The other important thing is, when the lipid soluble antioxidants, they go to the lipid environment, for example, let's say cell membrane, and they are doing antioxidation there. The way these antioxidants work is that, imagine I am an antioxidant and there is something, let's say this is a lipid and this has an extra electron on it. So this is oxidized and it is all upset and it is shivering with anger and it's going to attack others. And I am an antioxidant. I'll go and I'll take a hydrogen from here and then I'll combine with oxygen. I will become oxidized. I can handle that pain better than this guy. So I will cure it and I'll take the pain. But now I am oxidized. I need to be cured as well. So vitamin E will become oxidized inside the lipid membranes. Then it would come near the door of the lipid surface. It would come at what we say the interface between the water and the lipid area. It would come there and it will need some water soluble antioxidant to heal it. So imagine it, I am that lipid, I'm vitamin E, and I'm holding fire in my hand now, which I've taken from a lipid. I need to go to some water soluble interface and say to vitamin C that, hey, guy, can you take this and make me recovered so I can go back to the fight? So then vitamin C has to come and take that electron from me, and I will become restored and I'll go back and start fighting again. And vitamin C would now neutralize it. Now you could ask that, hey, who would help vitamin C now? Glutathione helps vitamin C. So glutathione can restore vitamin C by taking the oxidation from it or the electron from it. Then glutathione works with the NADPH system to use that energy into metabolic pathways. So when we talk about anti-inflammatory aspects of the food, omegas should be the first one to be looked at. And I'll give you a proof of that. When you go to a doctor for inflammation, what do they start you with? Ibuprofen, aspirin. What are these? Ibuprofen is a cyclooxygenase pathway blocker, which is the arachidonic acid pathway blocker, which is an omega-6 pathway blocker, right? Aspirin also cyclooxygenase pathway blocker. Most of these drugs, the anti-inflammatory drugs, other than the opioids, the most of anti-inflammatory drugs that we use are actually cyclooxygenase non-selective or selective blockers. That is a arachidonic acid pathway. So all of our anti-inflammatory, all of our painkillers, all of our antipyretics, anti-fever are actually arachidonic acid pathway modulators. So the first thing to focus on will be arachidonic acid, which means omega-6. Arachidonic acid comes from omega-6. And to counter the arachidonic acid is omega-3. So that's the first one. Then are the antioxidants. You can have them. Antioxidants are present in berries. They are very, very good. And they are very rich in antioxidants as well. Then vitamin C, vitamin E, whichever foods you feel. That is the start. Curcumin, I think, should always be added. So these are the molecules that one should start with. And then you modulate it. Then you see, and of course, with your doctor's advice, because some of these can cause blood thinning. And if somebody is already on a blood thinner, then this can compound it. So talk with the doctor, or you do your research. There is a good news here. I'm going to go away from mechanism for a second. My team has created an AI-based system where they are feeding the transcripts of my videos. And you can go to that AI system and you can discuss with it that where did Mubin discuss this and where did he discuss that. The AI not only discusses it with you to say Mubin discussed, for example, today's discussion will be fed into that system. And then you can talk about it with, in this discussion, what did he talk about foods for inflammation. Not only that, it would actually give you the reference to the video to say he discussed this in this video. I have, I think, about 2,000 videos now discussing various mechanisms in these little sound bites, which are embedded, almost buried in these long chit chats. And people don't watch these chit chats because they're, number one, long, and number two, they don't know where is the area of interest for them. Where will it be? Is it at 1 hour, half hour? So that AI system could actually help with that as well. So going back to the foods, antioxidants are very important. Then I would actually expand that to say more than the foods, near infrared light, that is morning walk, where the sun is at an angle, or evening walk, where the sun is an angle. Not that sun has gone away, but sun is still out there, but it is an angle, not at the zenith. That has a lot of near infrared light in it. It is free. Plus, if you walk, You are becoming healthy. Plus, if you walk near the trees and plants, then you even become healthy more because of the plant um, essential oils also reduce stress. Near infrared light can penetrate 4 inches into our body. It can penetrate through the clothing that we wear. So you could be all bundled up and go in the morning walk and receive near infrared light for 10-15 minutes, and that would heal mitochondria. Mitochondria are the first victim of inflammation. They start getting damaged and fragmented, and they do not work correctly, which produces a lot of reactive oxygen species, which causes all hell breaks loose within the cell. So, recovering mitochondria, simplest, near infrared light. Methylene blue is another that helps recover mitochondria. Uh, so, I would start from these, and then you can start modulating and adding or removing. So Sigrid says, will that work under cloud? So it really depends, that as long as you get some sunlight. Sita says that yellow time before sunset near infrared. Correct. Similarly, that kind of colorful time in the morning as well. Janet says, no one feels bad for the towel rack. Did you notice? Um, Jim says, even in North Iowa, there is snow up here. So as long as there is sun, if there is sun, then even if you're bundled up in the clothes, warm clothes, the near infrared can still penetrate. And let me make it better. If near infrared is not available, then a near infrared lamp. What happens is, when you go to Amazon like place to buy a near infrared lamp, there are mixed lamps that are sold, which are infrared and near infrared together, because people do not believe in a lamp that doesn't cause heat. So they kind of sneak in the heat generating wavelengths as well, or near infrared wavelengths too. But these are not good. The near infrared is used from 10 to 15 minutes. Infrared, the heat one, needs to be used for 20 minutes. So they both are needed at a different time. There are a little more expensive, maybe $60, $100 or so, where you can switch between near infrared and infrared. And you could have those that, let's say you use near infrared for 10-15 minutes in the morning or evening. You can use it as many times as you like, but at one time, don't go beyond 15 minutes. And then you can have sessions with the infrared, which is heat generating, for about 15 to 20 minutes. Saunas are very good. Saunas activate heat shock proteins. Heat shock proteins activate autophagy and cell healing and clearing. So saunas are very good, again, within the good timeline, without kind of burning out the cells. Saunas are good. Cold baths are very good because they also activate heat shock proteins. So there are so many ways that we can help our cells be less stressed. Critical Thought says even green leaves can reflect lower frequency light. Very good. Nipa says, yesterday I bought Himalayan rock lamp. That's a great feeling. Yes. And the lamp is beautiful as well. Margaret says, all of these are on my grocery list. Very good. Wookie says, ultraviolet radiation is best, but I burn easy, so I try for early morning or late afternoon sunlight. The rare times I am outdoors, I prefer less heat for almost everything. Even eat my food lukewarm. That's fine. Retro Plank says, is this live? Yes. So far it is live. Then it will become recorded in a few minutes. Do hot tubs activate? Yes. So heat shock proteins are called heat shock proteins because the scientists who were working with the cells, they were warming them to just see what do the cell do. And so what they do is they warmed the cell, or they heated the cell, and they looked into the cells to see what is going on, and they found a new set of proteins that were produced. So it's nothing surprising when our cell is under various environments. It will react to those environments, healthy or unhealthy environment, by the nucleus opening up the genes according to the functional need of the cell. Messenger RNA would come out that would help make proteins. Those proteins would do their function. So the cell was heated, or cell were heated in lab, and a new set of proteins came up. And the scientists call them heat shock proteins. Afterwards, it turned out that heat shock proteins are not only produced because of heat. You could be under the sun, you could be in the hot water tub, you could be taking a shower, you could be in a sauna, not only with the heat, but with the cold as well. Because these are really the stress proteins. They're not heat shock proteins, they're just shock proteins. You shock the cell with exercise, you shock the cell with the temperature, cold or hot, you shock the cell accidentally with other stressors, or unknowingly or unwantedly, the heat shock proteins will come into being and they'll work. Melatonin is very, very important as well. Melatonin is actually an antioxidant as well, which is a water soluble and lipid soluble antioxidant. So it can actually work in both environments, just like alpha lipoic acid, which comes from the omega 3s. That is also an antioxidant for water soluble environment and lipid soluble environment. It can work in both environments. Jim says, I have a 6-foot tall UVB light. Very good. I'm jealous. So Hilary said that I think that a near infrared or red light directed towards one part of the body will help other parts as well. So no, so the near infrared light, when penetrated to our system, what it does is it actually bounces. Have you seen in the old time, there were those, maybe even now as well, there are those little lamps which have these strings, plastic strings, and you have a bulb that is lit up in the center, and then those plastics and their ends are all shiny. Our blood vessels are that way as well. So when the near infrared light enters our tissues, it kind of bounces around in our blood vessels and spreads. So it doesn't spread a lot, but it spreads in the local vicinity. Interesting thing is that because it can penetrate up to 4 inches, so imagine you have near infrared light on the back, you would go almost half of the body. Then if you put it in the front, that would also go half of the body. So it is pretty easy for it to penetrate and then spread a little, but it cannot spread. For example, if I have near infrared light on my lungs, that would not help my knee joint. It would, if it reduces inflammation and inflammatory molecules are not going in the other places, that is a indirect effect, but not direct effect. Kate says, yes, love those lamps. These are very important lamps. Or morning, and again, it's not necessary that we have to spend money. Morning and evening walks near the plants, that all is very good. So Hilary says, each time 15 minutes front and back. Yes, 15 minutes max. So 10 to 15. Below 10 minutes, it doesn't work very well. And after 15 minutes, it doesn't work very well. So 10 to 15 minutes is the window which I discussed earlier as well. That was a study. Janet says, we are live making fun of AI. Thank you very much. That AI is really cool. Janet says the viscosity is changed inside of the mitochondria, so more ATP. Well, here is the thing with the infrared light and the mitochondria. Just like plants have chlorophyll, which interacts with the light and then can take energy from there, and the plant can make their ATP from that. Mitochondria complex 4 is a it's a cytochrome enzyme. It is a chromatic enzyme. It reacts to light as well. The only thing is, other lights cannot reach inside the cell that well, but near infrared can. Mitochondria's complex 4 can actually pick up that light and start working with the ATP synthase and start activating the mitochondrial function. This is something, if you like, I can very quickly draw and explain why the um, light is so interesting for mitochondria. So mitochondria has 4 complex electron transport chain. Inside the mitochondria has 4 complexes. I'm just going to make them like this: complex 1, 2, complex 3, complex 4. And then there is a little motor that helps make ATP called ATP synthase. ATP synthase. And these are complex 1, 2, 3, and 4. I have discussed this many times. I don't want to take too much of your time discussing how the electron transport chain works. Generally, the energy, the fuels that you eat, carbohydrates, lipids, proteins, these fuels, their energy is extracted and loaded on a couple of donkeys. These donkeys are NADH and FADH2. So these are molecules that are carrying that energy. They bring that energy in here, and that energy is then passed through this assembly line, and energy is harvested from it, which would then allow hydrogens to go out. Then hydrogens will come back from here, ATP will form. That's fine. When our mitochondrial electron transport chain is under stress, what happens is that the chain is not working correctly anymore. So it is not taking up the nutrients very well. It is not passing them through the system very well. And imagine that, let's say, the complex 3 isn't working correctly. Or let's say the vehicle between the complexes, for example, CoQ10 is a vehicle that takes electrons from complex 2 and bring them to complex 3. Imagine we are deficient on CoQ10. Where did the CoQ10 go? We were in an inflammatory state. We were making a lot of reactive oxygen species. CoQ10 is a mitochondrial antioxidant, and it started picking up those it is antioxidant plus it's a vehicle within the electron transport chain. So it started picking up the reactive oxygen species, and it got dead in that process. It got consumed in that process, and now there is a lesser amount of CoQ10. So when there is less amount of CoQ10, the electron passage from complex 2 to 3 isn't working very well. And so the energy that is coming in is leaking out from here in the form of reactive oxygen species and then causing damage. We desperately need this mitochondria to start working again. We want this assembly line to become okay again and pass the energy forward here. So guess what we can do? If we shine, so now for everything to reach here, for example, melatonin, or for example, CoQ10, for these things to reach here, the mitochondria has to be accessible. Imagine that the mitochondria is sitting in a cell which has a blood supply that is obstructed. What are you going to do to this mitochondria, or obstructed or reduced? You are giving melatonin, but that isn't reaching the cell in sufficient quantity. You're giving CoQ10, but that isn't reaching the cell in sufficient quantities. What are you going to do? So what you do is you give near infrared light. Near infrared light can shine on complex 4. Complex 4 is a chromatic enzyme. It can pick up the energy from light and start performing its function of moving hydrogen out, which would then come back from here and ATP will be formed. So just these two would start functioning. And as soon as the ATP is formed, the cell would start getting energy to start functioning correctly. That would start healing mitochondria as well. That would start healing the cell as well. And this chain would start working as well. So sometimes for mitochondrial help, if we cannot reach it through the highways, then you reach it through the space by giving it near infrared light. Complex 4 is very, very interesting. It also has nitric oxide attached to it. Some literature says that complex 4 manufactures nitric oxide. Some literature says that it traps the nitric oxide that is present in the environment and it holds it like a magnet. And then complex 4 would understand how much energy is passing through the system. And based on that, it can release the nitric oxide in the environment to cause the blood vessels to dilate and bring in more nutrition here. So when you shine near infrared light on complex 4, the complex 4 would release nitric oxide, which would then dilate the blood vessels and improve the supply to that cells as well. So not only you restart the electron transport chain, which starts making mitochondria healthy, but you also help produce or release nitric oxide, which would improve the blood flow to that tissue, which is being damaged, and you have shined near infrared light on it. So I hope that makes sense. Every single topic of these is hours long discussion. I'm just kind of giving you high level pointers. So Hobby E-Waste says, if all the iron was taken out of the bloodstream, would it become a magnet? So for iron to become a magnet, it has to be aligned. The atoms have to be aligned in a specific way. So not all iron is magnet. However, the question is, how much iron do we have at one time? I'm going to Google it. How much quantity of iron is present in a healthy adult? So 3 to 4 grams of iron in their body. So yeah, if you can somehow pull out that 3 to 4 grams of iron, of course the person would die. That is 3 to 4 gram. Then if you magnetize it by making the atoms in the right direction, then it will become magnet as well. Paul says, thanks. It is good to see a review of more in-depth teachings than before. Thank you very much, Paul. Cool. So what else before we break? If we don't have any other question, then we break. But please see that omega-6 less, omega-3 more, the morning walks or the walks under the rising sun or setting sun or near infrared lamps, antioxidants, both lipid-based and water-based, being near the plants. Essential oils from plants are very, very relaxing. They reduce the corticosteroids, they reduce epinephrine levels, and they strengthen natural killer cells. That is forest bathing. So Terrace Saramanga says, how do you remember all this stuff? So I say to my medical colleagues who also ask me this, that I have taken one for the team and I spent the whole day in the books and studies. So I just keep collecting this to present it back to you. So that is my sacrifice, if you can say, that I'm aloof from a lot of the environment around me and activities. I don't have a social life as you may have, and my focus is on studies. I tell my medical friends that I read all those studies which you will not read because you don't have time, and then you can discuss it with me. Interestingly, not everybody is interested in discussing these things. Janet says it is hard work. It doesn't just have, it is hard work. My wife sometimes looks at me, and at night I have the phone on, and she hates it when the phone is on. But I'm reading some study, or I have a question in my mind, and I'm Googling it and finding studies and looking at the mechanisms. So it's just that I just keep working on it. Eugene says Dr. Bean should be bottled and distributed worldwide. Heather says, thanks, Bean, for all your work. That is helping all of us. You're very welcome. We are in a more stressed out environment since COVID. So there were people who got long COVID. There were people who got vaccine injury. then the lockdowns and the financial aspects of this, then the divisiveness that appeared. um, there is a lot of stress. And I think every one of us, even if we think we are not inflamed, if we think we are not inflamed and we want to prove it before we manage it, then have a trend of CRP. CRP is the most important and easiest way to understand if there is inflammation or not. So CRP levels, one time level, are not enough to do, because maybe I have some infection going on right now and my CRP went up. So if I do a test right now, it doesn't mean anything. It just says at this time I'm inflamed. But if you look at the trend over, let's say every 3 months you do your CRP, every 6 months you do your CRP, and you see that there is a trend that CRP is higher and stays higher or continues to go up or fluctuates but is higher, then you have inflammation. And that inflammation needs to be controlled before it becomes diabetes, before it causes neuropathic changes, before it causes cognitive declines, before it causes muscle fatigues and joint pains and liver damage. Um, and I believe that, and I've done these discussions, there are studies for that. For example, diabetes, type 2 diabetes mellitus, if you control inflammation and inflammatory foods, it reverses. So it is important for us at this time, the whole world, or at least I can speak about US, we got damaged. We are scarred. And we are scarred in so many ways, not just the COVID or vaccine, the finances and the divisiveness and the stresses of the environment, and then the AI's emergence, causing problem with the jobs and so much more. We are under stress. And for that stress, the first thing to look at is inflammation. And what I find interesting is that managing this is actually cheap. Intermittent fasting is cheap. It is money saving. Going out for a walk is healthy and cheap, right? So mitochondria becomes restarted with infrared light. Body's autophagy restarts with the hunger. So cheap ways and healthy ways to become healthy. Hobby e-waste says, so we have an iron blanket of around a millionth of an inch if there's only a few grams spread out. Very interesting. Thank you very much for this. Kate says, you're truly appreciated around the world. I've met so many people who know you. Thanks a million. MCAS from wax injury makes CRP high for me. This info helps. Yeah, so if CRP is high, the basic understanding from the high CRP, C-reactive protein, means there is something in our body somewhere or throughout that is continuously telling liver to produce CRP. Liver receives that message in the form of interleukin 1 and interleukin 6. Interleukin 1 and interleukin 6 are produced in the area, in the tissues which are inflamed, and by the immune cells that are fighting there. Now, this could be memory B cells sitting in the bone marrow, just willy-nilly producing these inflammatory molecules, or it could be an actual problem somewhere where this is happening. So when the liver receives interleukin 1, interleukin 6, tumor necrosis factor, it responds by helping making various proteins that would go and fight as well. The kind of proteins it makes are, for example, C-reactive protein is one that would go to the area of the damage. It would spread out throughout the body. Wherever the damage is, naturally it would go there as well. And it would then start coating the damaged cells and coating the bacteria or pathogens or viruses or fungi and try to help immune system kill them. So that is C-reactive protein. Then the liver produces hepcidin, liver produces ferritin, liver produces complement system. So liver produces a bunch of proteins to help with the fight. This all is called innate arm system. These are the proteins of the innate arm. We know the cells of the innate arm, like macrophage and dendritic cell and natural killer cells and neutrophils. The liver acts as a partner in innate arm as well by producing acute phase reactants. These acute phase reactants, CRP is one of them. So because CRP is always increased when there is inflammation, whatever kind of inflammation, whatever reason for the inflammation. It is easier to figure it out. And presence of CRP, continuous presence of CRP, means continuous inflammation. That means it is paramount for us to control the inflammation while we're trying to figure out why is it happening and where is it happening. I've seen sometimes physicians who are saying that I'm going to not control inflammation other than if it is causing pains, and we would try to see where the problem is. No, the inflammation itself is a problem as well. So while we're trying to figure out where and why and when, we have to control inflammation so body is not damaged. Um, Eugene says, Luffy has stopped complaining, maybe fell asleep. I heard my son going down. So he may have opened the front door and Luffy may have gone out. I have to go and find him to say, come back. It is, but it's not raining as well. So maybe he went out from the back side. Stevie says, awesome. Thank you so much. You're very welcome. Janet says, Dr. Bean's only imperfection is that he licks pencils every time he finds himself in a post office. That was crazy. And I got COVID. Sita says, I thought inflammation is beneficial as at first. Absolutely. So this is why acute inflammation, don't try to get ahead of that, because acute inflammation means we are fighting something and this is needed. The chronic inflammation or subclinical inflammation are the ones that we have to get ahead of them. That is why I said CRP trends. That is why I said if for example, somebody who has diabetes and the diabetes need continuous help, that means they have continuous inflammation somewhere. Somebody who's developing liver damage or the renal damage or the cognitive decline or the Alzheimer or dementia-like situation or muscular fatigues, they actually have inflammation going on. This idea that we are just becoming old and our old cell are not functioning correctly. I have answered this idea many times before. Our cells are always young. We are continuing to make a new cell membrane all the time. So if this is a cell, the cell membrane is being made all the time. It is being refreshed and renewed all the time. Within the cell, the compartments, for example, Golgi apparatus, mitochondria, endoplasmic reticulum, smooth endosomes, peroxisomes, their membranes are being rebuilt all the time. The enzymes that are working, they are made, they do their function, then they're torn down by recycling or ubiquitination, and then when they're needed again, we make them again and they do their function. Same way messenger RNA, when we need it for something, it is made, it does its function, it is torn down within 2 to 3 days. The DNA, when a gene opens up, then the gene closes down. So everything is new. The only thing inside the cell that is bad is the misfolded protein aggregates. Those misfolded protein aggregates make the cell's functions bad. So then you could challenge me to say, okay, does that mean that if I do autophagy and intermittent fasting, all of a sudden I'll be just brand new young? There is one problem, and that is epigenetic changes. And epigenetic changes do not just come into being that, hey, there is an internal clock going on and our cells have to have a problem. No, epigenetic changes at some point. Imagine you yourself at a baby stage. When you are moving forward in age and you are eating carbohydrates and other things and the misfolded protein aggregates are forming, at some point, there will be sufficient quantity. That may be at 20 years or 30 years or 40 years or 50 years, depends upon the person's environment, how stressful, because that contributes to the reactive oxygen species, plus what kind of foods. Once there is sufficient piling up of the garbage, the misfolded protein aggregates, that is when DNA repair enzymes do not work correctly. That is when mutations in the DNA are kept there because the enzymes are not able to repair them. Now we have epigenetic changes, environmental changes in the DNA, which then causes bad proteins formation. So now we have damaged the manufacturing units and we have damaged the recipes. And so when those recipes give rise to new protein, they're already damaged. So what do we do about that? We start doing autophagy, we start emptying the cell as much as we can and hope that our DNA repair enzymes will then become corrected and they would start removing the epigenetic changes and will become healthier, energetic, and younger. There are other reasons for epigenetic changes. For example, light shining on our cells would cause epigenetic changes, or DNA repair problems, which, if not fixed, will cause epigenetic changes. Chemicals can do that, pollutants can do that. So it's not just always the food. There is a lot of things that can cause the DNA damage. The point is, we should be able to handle it. Now, for epigenetic changes, I would concede that sometimes, even if our cell is healthy, our cell decides to not repair a gene. The reason for that is going over the whole DNA length and looking at every single gene and correcting this is a very expensive and time consuming and energy demanding process. So many times our nucleus just doesn't get to the genes to fix them, and that can accumulate. But if we add to the problem by having protein aggregates, then we have compounded. Maybe if we just wait for the epigenetic changes to occur because of environmental factors and not have inflamed cells, we may be actually living healthy for 200 years. We have accelerated that time downwards because we have extra cellular debris present in there. Terry Saramanga says, "Where were you when I was 10?" It is actually so interesting that youngsters do not want to hear this. I have, I was speaking with a youngster and I was trying to say that if you take care of yourself now, you will actually live healthy and longer. And he kept saying, I know that I should eat healthy foods. I said, but if you knew the mechanisms, you will know which healthy foods. He said, as long as we are on the same agreement that I should eat healthy foods, I don't need to know the mechanisms. So I failed to convince a youngster to understand how to have a healthier life. Marcy says, but we learned from Dr. Bean never to lick or touch writing implements in public places. Thank you very much. Yes, I was the lab. So this is the discussion for today. Paul says, when I spent 4 years in Michigan, it's a great day if you don't know and learn to appreciate that saying. Correct. So I haven't even touched on the glycation or the fried foods and how do they. So the question about the inflammation, zero fried foods, zero superheated foods, processed foods. And it's not that I'm against some commenters say that Mubin is against fried foods and he doesn't understand that the oils are the problem. I have given this answer many times that hemoglobin A1C in our body is hemoglobin with glycation on it, with glucose is attached to it, unnecessary, incorrectly attached to it. That happens at our normal body temperature. There is no oil here. It is the heat which actually causes the deshaping of the molecules. Yes, bad oil would help compound that problem. Yes, presence of lot of glucoses in the environment will help compound that problem. Yes, peroxide lipids are totally dangerous. So oils are to be blamed as well, but heat is to be blamed as well. So superheated foods that are in the processed foods, they heat them so much that they kind of clean them up so that they can be preserved for a longer time. That de-shapes the molecules, which these age the advanced glycation end products, which we when consume, they wreak havoc on our GIT. They make the GIT permeable, they make the GIT damaged, they cause inflammation in the GIT. Then when the GIT becomes permeable, they can get absorbed from those leaky places back into the blood circulation, and from there they can go to the other places. Interestingly, medical community does not believe in leaky gut, but they do believe in inflamed epithelial barrier of the GIT. So if you say, don't say leaky gut, and you say, I may have inflamed gastrointestinal epithelium, they might agree with that. And they say, okay, yeah, we should look at the microbiome, and we should look at what you're eating, and we should look at what kind of inflammation is present. So glycation and products have their own way of damaging our cells. They're the most horrible things, even more horrible than omega 6s. Paul has given the best message. Remember to like the button on the way out. So, correct. Critical thought says Millard reaction. Correct. That is a reaction. Dr. B loves, oh, yes, I do actually love French fries. Every time my son buys a burger, I get the French fries. I love them. And I have started reducing them. So, overheating the microwave. So, anytime you give extra energy to the system, microwave or oven or whichever way, if it becomes hot, then that would cause de-shaping of the molecules. And as soon as the molecules are out of shape, they are going to cause damage to our GIT. And if they would cause the damage to our GIT, it is interesting though. There is this very common thought that the advanced glycation and products, or these misshapen molecules, cannot enter our body because they are broken down by our digestive system before. And that is not true. The misfolded proteins, to give you an example, take an egg and fry it. Maybe not fry it with the oils, just heat it too much. It will become very hard little thing, correct? These are misfolded proteins in there. They are coagulated proteins and substances in there. Our body cannot handle them very well. We cannot digest them very well. So it is not necessary. And there are studies to show how advanced glycation and products that we eat in the food end up in the blood. The normal healthy food, we can break it down. But the advanced glycation and products can actually bypass our digestive system because digestive system doesn't know what to do with them. And then they take a hitchhiking. They steal the pathways for other molecules to enter our body, and they just kind of tag along and enter through them. Then they cause the permeability change in our GIT, and then from the leaky spaces, they enter into our body as well. There are so many mean ways to enter in our blood circulation. So this thought that somehow I took fried foods and they would have been taken care of by our digestive system, and we have broken them up and they are into smaller pieces and these pieces are harmless. No. Ask someone who after food gets inflamed and see what's going on with them. This is so correct. The youth is wasted on the young. Best sea salt and finest apple cider vinegar with the fries, of course. So this is the discussion. Tomorrow morning, 7 a.m., we'll have a morning walk. So what we have is a Zoom link that I think my team publishes every week. And then we have a meeting, we do a walk, and we talk and we have fun. So thank you very much. And I would... Very good. You're very welcome. So thank you. Have a good night. Thank you very much for hanging out with me at this late time for some, and even very early for others, and middle of the night for some. Thank you. And please like, subscribe, and share. That is the way you can help me back. And I would see you guys from the next week as well, but tomorrow, 7 a.m. Pacific. Bye for now.